Welcome back to our topic of gene expression. So in this series of videos we're going to be talking a lot about gene expression, and we're going to be talking about the central dogma of molecular biology, which basically says that we start out with DNA that's in the nucleus, and DNA essentially is the permanent carrier of genetic information in eukaryotic cells. And we can do a process known as transcription, in which we can take a DNA template and we can synthesize a new kind of nucleotide polymer known as RNA. And we mentioned in other videos that there's different types of RNA. For example, we have messenger RNA. We also have another type called transfer RNA. And we also have another type which we talk about in the peptidyl transferase video called ribosomal RNA, and there's some other ones that are minor in nature that we don't really talk about called small nuclear RNA. Suffice it to say that one is used by the spliceosome when you're trying to splice out introns and glue the exons together during mRNA processing. Okay, and then we can essentially use these first three right here, and we can take the RNA and do a process called translation, and translation essentially is carried out by an enzyme known as the ribosome, and we can get the final product of gene expression, which is our protein. So basically what gene expression is it's going ultimately and indirectly from DNA to protein. Okay, and the proteins could be things like fibrous proteins that give structure to the cell and to the body, or we could have things like globular proteins that include enzymes and antibodies would be for things like defense, and enzymes would be for catalysis. Okay, so we have to do gene expression to get those products. We also have some intermediate processes that occur. For example, when we synthesize things like mRNA, we have to do something called mRNA processing. So for example, if we're synthesizing mature mRNA, we're going to have to do 5'7-methylguanosine capping. We're going to have to do intron splicing, where we splice out the introns and glue the exons together. We're of course going to have to use an endonuclease to cleave off a component of the nucleotides towards the 3' end, and then we're going to use an enzyme called polyadenylate polymerase to essentially add adenosine monophosphate residues to the 3' end. Okay, so we've got to do a little bit of work once we actually do the transcription process. We're also going to do some modifications of tRNA, like making funny bases and so forth. For instance, we can deaminate adenosine to make inosine. So we can do various things to the tRNA to ultimately get mature RNAs. Okay. And like in the case of mRNA processing, we have a process in which we activate the tRNA, or we charge the tRNA, in other words. So what I can do is essentially I can have a tRNA, but the tRNA really by itself doesn't do anything. Okay? Really what T stands for is transfer. So what the function of tRNA essentially is it's to transfer a molecule known as an amino acid. Okay, so what we're essentially going to do through a process that we're going to talk about in this video and the previous video is we're going to charge the tRNA with an amino acid, and in the process that does something called activating the amino acid. This complex right here, or this, we could call it this conjugation between the tRNA and the amino acid, this is absolutely required for the mechanism of the ribosome. It turns out that the ribosome cannot directly deal with just simple amino acids. You've got to ligate it to the tRNA first, and only then can you deal with polymerizing the amino acids into a protein. And the general name for the type of enzyme that glues or ligates the amino acid to the tRNA is what's shown here. It's called an aminoacyl tRNA synthetase. Now I'm going to do a brief recap of what we talked about in the previous video, and I want to talk about the origin of the name synthetase. So the name synthetase in the name of an enzyme sort of gives us a clue as to what the substrates might be for that enzyme. In general, when you're talking about a synthetase, you're usually talking about an enzyme that glues two things together, usually using ATP, although sometimes GTP can be used. But in general, we're really talking about ATP, and certainly in this reaction, aminoacyl tRNA synthetase has an absolute requirement for adenosine triphosphate. Okay? And when you have synthetases, all synthetases are referred to as ligases. If you're looking at enzymatic nomenclature like the EC numbers, ligases are classified as class 6 enzymes. Okay, class 6, and all synthetases are ligases. We've actually seen another synthetase in the TCA cycle. It's called succinyl-CoA synthetase, and in the direction that the reaction is named for, it will actually use GTP. Okay, so that's an example of one that uses GTP, but in general, most of them use ATP. Okay. Another example of a synthetase is DNA ligase that's used in DNA replication. Okay, so those are just some examples. I also want to think about a really important concept known as rate-limiting nutrients for protein synthesis. And in general, there are 2 rate-limiting nutrients for protein synthesis. And those rate-limiting nutrients are known as, number 1, adenosine triphosphate, for obvious reasons, as we'll see soon, and then also amino acids. Okay. I want to think about why ATP is a rate-limiting nutrient. Well, as we'll see when we actually look at the mechanism of this enzyme, and hopefully you watched the previous video to see the mechanism of type 2 aminoacyl tRNA synthetases. But for every time we ligate one amino acid to one tRNA, so if we want to make this guy, we have to expend one ATP, which basically in the long term means the following. For every amino acid that we polymerize into a protein, we have to burn one ATP. The reason you have to burn the ATP has nothing to do directly with the ribosome. It has to do with the fact that the ribosome has a requirement for using the amino acid while it's bound to the tRNA. And the way you make this linkage right here, this linkage is to expend an ATP, and that's through the mechanism of aminoacyl tRNA synthetase. And obviously, per cycle of aminoacyl tRNA synthetase, you're of course using an amino acid per cycle. Okay, so basically per cycle, you're going to use one of all these guys, ATP and amino acids. Okay. And so these right here are basically the rate-limiting nutrients for protein synthesis. You could imagine that if we start doing a lot of protein synthesis, we're going to have to expend a lot of ATP. ATP is the energy currency in the cell, so we say that protein synthesis is a very energetically exhaustive process, but for obvious reasons, it's absolutely required for life. And that's also a good reason why you definitely want to make sure to eat your meals and get plenty of calories, because ultimately those nutrients are going to be degraded into different end products, but in the process you'll generate things like NADH and ubiquinol, which will power the respiratory chain of the mitochondria, leading to ATP synthesis by ATP synthase. And so if you expect there to be significant ATP production, it's a good idea to get your food and nutrients from your meals instead of relying on gluconeogenesis and ketone body synthesis. Okay, so hopefully that makes a little bit of sense. You need to get your ATP, and you need a lot of it if you want the rate of protein synthesis to be rather high. So if you're trying to heal something like a wound, if you're trying to build muscle, as in the case of people who want hypertrophy from the gym, it's a good idea to eat lots of food, because that ensures you get lots of ATP along with amino acids as well. And you can certainly go back to the previous video to learn a little bit more about that. But those are the rate-limiting nutrients of protein synthesis, and they're directly used by aminoacyl tRNA synthetase. Now, one of the things that we'll elucidate when we look at the mechanism of this particular enzyme is you essentially will have this tRNA, okay? You'll have this tRNA, and there's a reason I haven't drawn these hydrogens yet. Of course, this nitrogenous base will be adenine up here, and then you'll have basically the rest of the business going towards the 5' end up here. So this is basically the 3' end of the tRNA nucleotide polymer. Now, in general, there are 2 types of aminoacyl tRNA synthetases. Okay, there is one type of aminoacyl tRNA synthetase that's essentially going to ligate the amino acid onto the 3' hydroxyl group. So what you'll see essentially is something like this. So here will be the R group, and this will be the alpha amine. So notice that essentially the amino acid is ligated directly onto the 3' hydroxyl group. This is the 3'. This type of reaction where the amino acid is directly ligated onto the 3' hydroxyl group, this is called an aminoacyl tRNA synthetase, but this type is called class 2. And this was the topic of the previous video. Now there's another type of aminoacyl tRNA synthetases that's a little bit more complicated. Okay, and of course you still have the adenine nitrogenous base right here, and then the rest of this business goes off towards the 5' end of the tRNA nucleotide polymer. However, in the case of class 1, so we're talking about in this case class 1 tRNA synthetases, what essentially happens is you instead actually favor the addition of the amino acid actually onto the 2' hydroxyl group, which is actually really, it's a little bit intuitive as to why it happens, and to a large extent it's not really known. But essentially the amino acid gets put onto the 2' hydroxyl group instead of the 3'. And it turns out that the way the ribosome works is it's only good at dealing with these kind over here, where you have the amino acid ligated in an ester bond to the 3' hydroxyl group. So how do you get around the amino acid being ligated to the 2'? Well, the enzyme is going to have to do a little bit of work, and essentially what's going to happen is you're going to get a reaction known as a transesterification reaction. Okay? All a transesterification reaction is you take one ester bond and you essentially move that ester to an adjacent hydroxyl group. So essentially what's going to happen is this moiety right here, let me circle this acyl group right here, that group and this proton on the 3' hydroxyl group, they're essentially going to switch places. And that's part of the mechanism of tRNA synthetases of the class 1 type. And this mechanism is actually a little bit poorly understood, so we're just going to give a general mechanism as to probably how it happens. But the initial part of the mechanism is very well elucidated, and we're going to go over that now. So hopefully you have an understanding of the difference between class 2 and class 1 tRNA synthetases. In the last video we looked at class 2 because it's more simplistic. In this video we'll look at class 1 aminoacyl tRNA synthetases. So here we have a list of amino acids that are activated by the type 1 tRNA synthetases. This is, of course, an exhaustive list of them. I'll read them off to you. Arginine, cysteine, glutamate, glutamine, isoleucine, leucine, methionine, tyrosine, tryptophan, and valine. Okay. Out of these particular ones, I'm going to use glutamate as an example, just like I used alanine in the previous video for the type 2 tRNA synthetases. One thing you might find interesting to ponder is that when you look at the distribution and specificity of both types of tRNA synthetases, it's actually divided evenly and symmetrically. So 10 of the amino acids that are found in proteins are reacted with type 1 tRNA synthetases. The other 10 that are found in proteins are also reacted with type 2 synthetases. Okay, so it's an even symmetric distribution between the two types of enzymes, which is actually something that's actually pretty typical. Okay, so we're using glutamate as an example, and we're going to look at the mechanism as to how the type 1 tRNA synthetase works. I'll do the mechanistic steps in green. Like I said, these tRNA synthetases require this molecule, which is known as adenosine triphosphate. And the active nucleophile of glutamate is, of course, going to be this particular alpha carboxyl group. Notice also the glutamate has an extra carboxyl group in its R group. This is not the one we're concerned with. Always the nucleophile in this particular type of reaction is going to be the alpha carboxyl group. So what's going to happen is the alpha carboxyl group will do nucleophilic attack on the alpha phosphate of ATP. So if we're labeling the phosphates from proximal to the ribose ring to most distal, we have alpha, we have beta, and we have gamma. So gamma is most distal from the ribose ring of ATP. And when you do nucleophilic attack by the carboxyl group on the alpha phosphate, you're going to do something very similar to a nucleophilic acyl substitution that you saw in organic chemistry. This is referred to as a nucleophilic phosphoryl substitution. And since this moiety on ATP is already tetrahedral, you're going to generate something referred to as a trigonal bipyramidal intermediate, also referred to as a pentavalent intermediate. And I've, of course, eliminated the intermediate because it just complicates things, but suffice it to say what's going to happen is the phosphorus-oxygen double bond will reform, just like in the case of nucleophilic acyl substitutions, except you're dealing with carbon, and you'll get loss of a leaving group. So basically, the mechanistic steps are identical to what you see in nucleophilic acyl substitutions. Nucleophilic attack, generation of some kind of intermediate, collapsing back down to the pi electrons, and loss of a leaving group. And the leaving group, it turns out, in this case of the tRNA synthetase, is this moiety right here, which is referred to as pyrophosphate, and that's shown right here. Pyrophosphate has an interesting role in the cell. Essentially what's going to happen is pyrophosphate is going to react in an enzymatic reaction catalyzed by inorganic pyrophosphatase. Inorganic pyrophosphatase. This is a hydrolytic enzyme. Okay, hydrolytic enzyme, and what it's going to do is it's going to split the pyrophosphate apart using water, and so what you should end up with at the end of this reaction is something referred to as inorganic phosphate, which you'll find at physiological pH. You'll get two of them because you're splitting the pyrophosphate apart with water. Okay. This particular reaction is very important from the cell's work perspective because this reaction is associated with a delta G that is far less than 0, meaning it's very exergonic, and so it releases free energy that's used to complete this reaction and cause it to go to completion. Okay, so release of free energy is very important in synthetase reactions like this. Whenever you essentially do this kind of transfer, you essentially end up with this adenosine monophosphate moiety attached to, in this case, glutamate, although in the case of other reactions it could be other amino acids of those 10. So what you essentially have here is you have this, we'll call it this adenylyl group. I'm circling it in yellow. This is the adenylyl group. It's essentially an adenosine monophosphate AMP, and it's attached in a phosphoester bond to the remainder of this molecule, which is just known as glutamate. That's why this particular intermediate is known as adenylyl glutamate. Now here's the component where the tRNA becomes important. Okay, so what's essentially going to happen in this case, notice I can label the ribose ring. This is the 1', this is the 2', 3', 4', and 5'. I've labeled all the carbons of the ribose ring. The tRNA is going to come in. Of course, the 3' end, which is shown here, has the adenine as the nitrogenous base here. And what's essentially going to happen is the phosphate that is part of this adenylyl glutamate is going to essentially come over here and deprotonate the 2' hydroxyl group, which is essentially going to initiate nucleophilic attack by this set of electrons right here on the ester bond, the phosphoester bond of the adenylyl glutamate, generating a tetrahedral intermediate. Now once again, I've of course eliminating the structure of the tetrahedral intermediate, but you should know from organic chemistry that the tetrahedral intermediate will collapse back down to reform the carbonyl, and you'll get loss of a leaving group. In this case, the leaving group is adenosine monophosphate, and that leaving group is shown exiting right here. So this is the adenosine monophosphate leaving group. Now essentially what you have is you have the tRNA. Of course, I've grossly abbreviated the structure. I've designated that this part over here just goes toward the 5' end. And notice that we have this acyl bond, this ester bond that links the amino acid glutamate onto the 2' hydroxyl group of the ribose ring of the tRNA. Okay? And this is useless to the ribosome, so we're going to have to fix it. Now, this part of the mechanism is not well elucidated. But essentially what's going to happen is there's going to be a base somewhere in the active site. The base will deprotonate the 3' hydroxyl group, initiating nucleophilic attack on the ester bond of this aminoacyl group, generating a tetrahedral intermediate. In this case, I have shown the tetrahedral intermediate, and the negative charge will be stabilized by a positive charge in the active site. The tetrahedral intermediate will then collapse back down. And essentially what's going to happen is you're going to get loss of a leaving group, which in this case is the 2' hydroxyl group, as it comes out and abstracts a proton from that same base that was in the active site, and you regenerate the deprotonated state of the base. So now what I've successfully done is I've essentially transferred this acyl group, or it's an aminoacyl group because it's an amino acid. So this is an aminoacyl an aminoacyl group, I've successfully transferred it onto the 3' hydroxyl group. So once again, I can label these carbons 1', 2', 3', 4', and 5'. Now I've effectively transferred the aminoacyl group onto the 3' hydroxyl group, and this particular molecule, glutanyl tRNA, is now capable of reacting with the ribosome. So this right here, this is essentially your final product. And keep in mind that this particular type of reaction where we transfer the aminoacyl group from the 2' hydroxyl group onto the 3' hydroxyl group, this is referred to as a trans-esterification reaction. Okay, so hopefully this video gave you a little bit of intuition on the mechanism of type 1 aminoacyl tRNA synthetases. Go back and watch the videos on the type 2, and in the future we'll look at the mechanism of peptidyl transferase, the catalytic component of the ribosome. See you in the next video.